They're the transects.
Oh, and one of those white things you wanted to look at is
Oh,
up
we
there
can look
too.
at that. Oh yeah, those was like an urchin or
Oh yeah, one of those like porcupine urchins.
porcupine urchin.
Porcupine.
Not the official name.
There you go.
Not the
Zoom
official
in.
name, right?
Video, what were the segments?
Not a thing.
7, 8, and 9.
Oh, this is a reef-forming coral here.
Oh, that's a good sign.
Yeah. Look at this dude.
Very, very
Kind of
pretty.
coming
And we've
out.
entered into our color, our green, red.
Yeah, our little splotchy friend.
Mhm.
Get a photo of that. Sorry, I'm gabbing.
I like all the colors on it. Oh, that's the whole thing.
Yeah, it's very orange.
But not a sample.
I mean, we could
Okay.
sample it.
You think? Should we
Oh, there's something
cut a
in
piece?
there.
We got
Oh,
the coral clippers. Yeah, yeah.
Well, if you give it our coral expert
We could get
sample,
Yep, there's a
a
small
sample
little piece we could
requested,
get.
please, of this guy.
Okay.
And
Full
then
wide,
we'll
please.
grab one
Got
of
it.
the brown
And we'll
ones
do a
that we
brown
see
one,
a lot of.
and then the black coral. If we see one of those.
I think that's fine.
Okay.
Where would we like to put that sample, Science?
What do we got?
it is wide open everywhere.
Everywhere.
So, I mean, if it's going to be a small piece, like
Small
A, B,
piece,
C,
yeah.
D in the starboard or small
Yeah.
boxes, but
Yeah, maybe A.
we got plenty of room.
Just a small little portion goes in A. These are pretty hard, so they would be fine in those boxes.
Up in the top left-hand corner, it's another one of those urchins as well.
Yeah.
It's a good little spot.
I think we called them porcupine urchins.
Porcupine urchins.
We know that's not the name,
Unofficial.
but it just looks like a porcupine.
Just want to say a big thanks to Nav, pilots, video, and data logger for finishing up that transect. I know it takes a little extra concentration and time, so we appreciate it. Thank you guys so much.
Thank you, Kelsey, for being here.
Thank you for joining us, Kelsey. You're our interloper today.
I know, I like being y'all's interloper. It's fun.
Everyone loves a 12 to 4 shift so much that we always have a visitor.
It's a great 12. It's a do you great want me to shift. get to the box?
It's a very welcoming environment.
Yeah.
Oh, yes.
Second here.
And
Let's
part
see
of me
what's
thinks she
going
just
on
wants
there.
to make sure we do the
Good.
transects right.
Oh, I trust you.
Oh, sure.
Oh, sure. I just would feel guilty not being a part of it.
Trying to get an idea of what this guy could be. They turned, yeah.
Okay, zoom in on the Zeus, please.
Got it. Got more.
So just off the top.
Okay.
Yeah, that would be fine. Just where you, there's some orange color in. So that's a live tissue.
Just a little off the top.
Coral haircut.
Coral haircut.
No.
Wide cutting.
pretty hard, crumbly one.
Yeah.
So you're just going to try and take a bit? I feel like it's going to
Should we
crumple right up, or do you want to take the whole?
grab the whole thing?
Yeah, we can maybe just try to see what you can do if we take the whole thing. We take the whole thing, but
If we it's crumbly,
yeah,
so if
it
it's just
is
going to
gonna
fall apart.
Yeah.
Well, do we have the?
Can we slurp a piece? I saw a piece fall off. Is it possible to slurp it?
I would think it would be too hard.
Okay.
You would have to knock it around a little bit.
Well, we could try. I have seen them with the core where you kind of put it in and break it with the suction on. How big of a piece do we need? The polyps?
I wouldn't want to take the whole thing, just a couple polyps to show.
Okay, let's see.
Do you want to try
representative, yeah.
section?
Well, he's going to go for it.
That little piece, we can grab that.
We
if you
clipped
like.
it
you're
and
gonna grab
now
that.
grab it.
Yeah, wide, please.
Didn't
Yeah,
feel a thing.
All right.
I just didn't want to take the entire thing.
No, I
It's a
didn't
really
either.
small-
Bucket cams on
Small piece.
and readying the suction pump.
Yeah, that little piece fell at the bottom right there. Awesome.
Oh, unfortunately, I don't have the porch out. Bear with me.
You're good.
Annoying.
We're with you.
Okay, porch
No
out.
worries.
Roger, porch coming out.
At least it didn't fall all the way down.
Right.
No.
Then it would be
It's
gone.
all the way out
slow
now.
that valve down, please. Just
Roger.
take a good look. Check those before we use them.
Yeah.
Just to let you guys know, I promise we are working on the feed and the audio. A report has been sent in, and we hope to
Okay.
get you guys up running as soon as we can.
Yep.
What jar? We're putting in jar number 1, science?
Yep, that's good.
Roger.
How big of a piece? It's not going to be any issue with the size and sectioning.
I don't think so. It looks maybe
I don't
okay.
know. It's kind of borderline, isn't it?
Mm-hmm.
Let me see if I can just get it. Can we put it
You
in a
can
bin?
just try grabbing it, yeah.
Yeah. Zoom in there, please.
Got it. Are we going for the forward bin,
Whatever one they want.
It's.
science?
I think we were going to try to go for starboard A, B, C, or D because
Yeah,
they're smaller.
if it fits in one of those.
But if it's like precarious, we can put it in the forward.
Roger.
Oh,
Beautiful.
lovely.
Coming out.
Yeah,
Delicate.
they can go anywhere you want.
Yeah, let's put it maybe in A then, just because it's so small.
A in the starboard.
Do you want sample?
Yes, please. All that. Cameras, lights.
Roger. Box coming open.
Nice.
Awesome. Thank you.
Cool.
Box closing.
Data that was 082.
Yeah,
Yep.
and we might be able just to grab one of these other fans while we're sitting here. That would mean we wouldn't have to move anywhere else.
Yeah, let's do it.
Probably be easy.
Get our samples.
That way they could use it later for the transects.
Show me the arm and the bubble cam, would you?
Roger.
Just the jaws of it.
The jaws.
Yeah, the end of it. Thank you. Okay, let's look up. Oh, wrong camera.
Yep.
One of these brown ones?
Yeah, and honestly, you can grab the entire thing. There's plenty. I don't feel
where
as
would you like it?
conservative about these
Science?
guys. You can go ahead
Zoom
and put it
in on,
B.
hold on, I'll
There's-
get this, this one I got here.
Starboard box again.
Starboard box
Yeah.
B, please.
And
Roger.
zoom in on the Zeus as well, please.
Got it. Oh, I should have just left it open. Oh well. Here it comes again.
You never know, these coral could just jump right out.
Coming out.
Got enough of it there?
Yeah, I think that's great.
Okay. And B?
Yes,
B,
please.
yes, please. Where did the water go? Oh. Found it.
Stuff that right in there. I want to float away. All right, close it.
Box closing.
Awesome. Thank you so much. And we got to see the cool manipulator arm coral clippers. We haven't seen those before. It's for you, Shannon. You can go back and look at the footage and use that.
Yeah, I know. I was just thinking about that I need to,
Ready for dive salvo?
when I do highlight
Yep.
Roger.
checks, I'll find that.
Nice. All right, ready to carry on to waypoint 6?
Yes.
There was a question in the chat about if the
know about
viewers
that.
are able to
I would say
know
yes.
what these samples are after they've.
Not this one specifically. No,
After
I know
they've-
it's
Do you want me to bring the porch back in?
Yes, please.
Roger. Porch coming in.
Were we able to find the porcupine-looking sea
Porch
urchin?
is all the way in.
Those guys, did we find the name for that? Yesterday, we were looking for it, and I didn't see if we found what they were.
It looks like that one's like scooting over.
So Kelsey, we had a question that you were just about ready to talk about. How do people find out what the samples are identified as or information after the dive?
Yeah, so all the highlights from the dive are posted on the website. And then a lot of the samples that are collected are going to partner institutions, museums, and universities. But I'm not sure about the general public if they don't go and visit the curated collected site. Jeff or Sierra, do you all have any further insight about that?
I might have some insight. If you want my intake.
Oh, here's Bree joining us from the lounge.
Yeah, so the MCZ is where all the biological samples go.
Got it.
And they're like, if you just look up MCZ database, you literally could type in like Nautilus and then like in one of the search fields, and then you could see all the samples that have been collected from the Nautilus, and then you could see the years that they were collected and when they're what they're identified, and like you could even see like as years go by, different scientists come to identify things later, and you could see the updated identifications of things.
Well, Brianna, thank you so much. That is perfect. So for the viewers at home who might not know what NCZ stands for, would you mind giving us the full name?
Okay, full wide.
Got it.
Bouncing around all over
That
the
looks
place.
like an anemone sitting on it. I can't tell what that is.
Science, what were the letters of the boxes those went in?
That last one was
Last one was B, and the first
A
one
and
was
B.
A.
Okay. What is that?
What's that?
Whoa.
Whoa. Whoa.
So
Look
for
at
our
that.
viewers
Michael
at
says
home
cool
who
and
might
the
not
camera
know what
shifts over.
MCZ stands for, that is Museum of Comparative Zoology, and it is the Harvard Museum of Comparative Zoology. So all of the samples that have been identified and collected on the Nautilus cruises are available there.
These look like little. Now,
What do you think this is, Sierra?
That is a good question. I don't know what I'm looking at. It may be a
How do
kind of
I
looks
tag a
spongy
highlight?
to me.
Go ahead and zoom in.
It kind of looks
Got it.
sponge-like.
Wow.
See all the little
I agree.
Yeah, pores. It's like a tube sponge almost.
That's really cool. It looks fluffy and like
Yeah.
Zoom in a
furry.
little more.
I have never seen something like that. Yeah, it's
Try to get the.
yeah, I'm looking at the inside there. There's something chilling on it. Little crab.
Where's there something chilling?
Right down here. It's a little
Oh yeah.
fella.
There's one off to the right as well.
Oh yeah, there is.
Wow, what a curious little creature you are. Yeah, I would think some kind of sponge. Hard to say.
This is very interesting.
Okay, full wide.
Coming out.
That's all I got. So sorry, folks.
Oh, looks like another.
Do we need to get
little one.
any of those black corals?
It might be worth it.
Oh, there's another one.
Oh yeah, just because we haven't got any black corals collected yet.
Okay, so make the call. Is this one of those?
Yes,
here?
that's a
Should we?
Yeah, maybe the end of it.
Oh, and it has one here.
Dude, look
The
at
same
these
little anemone
sponge things.
Should we sample that?
We're pretty far behind, unfortunately. Do you mind?
Okay. Yeah.
Do you think you'll see it again?
Yeah, we've
Oh, yeah.
seen
I'll
some
just get up
earlier.
in front.
Sure, sure.
And then we'll have lots of time.
I think we'll have more opportunities.
Yeah. And yeah, there's like so many of these guys. What are you?
I won't put another step in.
I'm gonna look.
No, you can go ahead.
Oh, okay.
What do
Just
we
kidding.
They look like nice homes for
They do.
crabs and other things.
Come up on the winch.
Well, you know, they have
Okay.
those giant like barrel sponges in shallower waters. So it wouldn't be too far-fetched to think it might be a kind of sponge, but I've never seen one with that kind
Terrain
of
kind of peeking in. Yep. Pulling up on the winch there.
I'll look around.
There's more.
Yeah,
Look at all
those.
Oh, what's that like pinkish little ball?
Oh, yeah, that's sea urchin.
What's
maybe?
that?
That one.
think they're trying to catch up.
Someone said it looks
We're
like
okay, we can
melted
stop.
candy sponges.
Whoa.
Whoa.
There's one of those corals right there, right?
What is
Yeah.
that?
Oh,
Okay,
there
go
is
ahead
one
and
of
zoom in.
There's one of those sticopathies. Yeah, what it?
I would wish that it was
Is
like
that
a
an
snail, but-
anemone, like closed up?
Yeah, it might be.
Mhm.
It looks like an ancient, like Chinese vase.
It's got a cool pattern to it, yeah.
Wow.
That's
Really
pretty.
Yeah, there is that black coral right there. If we have a chance, we can just snatch that one up.
The little one behind.
Yeah.
Here,
It's
The
green.
question
That is
mark?
really cool.
Yeah, it is.
Keep you from having to
The
hunt
question
down
mark?
some more.
Yeah.
Coming out.
there will be more of them
High
if you
delta with
want
turn
to
on the 20.
Yeah, if you want
reposition.
to wait.
Yeah, that's what happened there. I got dragged. What's going on here? Okay. Sorry, I'll have to
You're
get in
good.
a better position there.
No worries. Yeah, I did see some more scleractinians.
Whoa. Oh.
Yeah,
What do you think all that white is?
I think it's one of those sponge things.
sponges. I don't know my shallow water sponge
That's
friends.
another one
Oh,
of
we've
those
got more of
closed
those
Whoa.
anemone
We're moving.
things.
Yeah, I think we're just trying to catch up.
Those sponges are cool.
They are really
looking.
cool, huh?
I like the one with the question mark and the
Yeah.
Yeah, you like
The
Yeah,
way-
now they're all gone.
circular.
We've definitely changed a lot in the last
Yes,
little bit.
I will tell everybody I spoke with the other watch leads and science leads that if we get to the top and we see everything we need to see, it's fine for us to come off and head back down.
Okay.
So
Oh, okay.
we're not completely locked into never coming back to this water depth if we have time in the dive and the top of the seamount is,
This angle is very
we feel thoroughly characterized.
tricky.
Right.
I feel like the perspective shows how steep it is.
Yeah.
That sonar. Yeah.
That's sonar is
yeah.
right. That's a lot of, oh, that's us, we're changing our
Are
heading.
you able to pull
and
that up, Maren, so the audience can see?
Yes, I should be.
You can kind of see
If they look at
it's
sat
changing
3.
depending on which way we're facing.
Yeah, it does.
We really are climbing a mountain.
Yeah, we've come to the near vertical. Are we dealing with currents yet at all?
No,
Oh,
not
good.
really.
Good.
Another urchin there.
Yeah,
It's
there's
a little bit now, actually. You can see it. Yeah. Okay.
If
Coming
you're
from the
a-
west.
If you're a climber, is this
Yeah,
what
that
you
is
call
new though. We
the
didn't have it a
crux?
few minutes ago.
Well, I was imagining
Yeah.
we're hitting that water depth
Nice.
Yeah.
where
Where are we
where
at?
the current was bad around this time yesterday.
We just broke the 200-meter mark, and isn't that the depth limit for the
Photic? Or-
yeah?
Well, we're definitely reaching into that, maybe the edge of the mesophotic.
Seems like people are starting to report that our video feed is returning to normal,
Okay.
Oh,
Yay.
and they want to thank the technical staff and helpers.
Yeah,
Thank you.
shout out.
They
Thank
work-
you.
Thank you.
They work very, very
They
hard.
make this amazing live stream possible.
Wow, Atalanta's got some light.
Yeah.
Oh yeah.
I'm going to close the iris just a little bit.
Roger. Oh, I'll wait on it. Never mind.
I forgot what it is. What is CCA again?
Crustoids coralline algae.
Okay.
Is everybody chiming off with it?
Yes,
Yep.
yes, and I never remember what that one is. Oh, what's that? Is that a big sponge
Another
again?
Another
Have we-
sponge.
Have we
Sponge
identified
castle.
this?
I'm
What is
content with calling it a demo sponge.
Okay.
Sponges getting sponges down to species again is also difficult.
What's this?
Type of sponge, maybe. Oh, Mariana, maybe this is it.
Jeff just reached over and pointed to something
Well, it kind of
random.
looked like it.
Oh, a little bit. Could be a type.
Atalanta
256.
bottle,
Well, we're
40.6.
lady.
Spongiopsoritis.
past the
Sorry,
20-meter mark.
Sorry.
Oh, wow. Look at all
Atalanta
that
main bottles at
orange,
40.6.
Isabella.
That's fine.
Train's peaking past the
Can we
20.
get a
Yep.
Just normal.
zoom in closer on the wall?
Coming on up.
Sure, go ahead.
All right. Zooming.
What
Doing a
are
full
these
zoom.
little guys?
I think this is the
Whoa.
neon glow they were talking about.
Oh, the neon glow.
These look
Cool.
like some corals.
Little coral, little polyps.
Yeah,
There's a lot of them.
they're orange.
Yeah.
Oh, very cool.
Coming out a little
Yeah.
bit.
Yay, the feed, we're getting, yes, more indications
'Kay,
from the
pulling
chat
out.
that the feed's better.
Great. All right, that's gauges.
Roger.
I'll log this as a possibly, because this looks kind
Someone said possibly, oh no, they're joking. Sandcastle sponge.
of dazzle sponge.
I was
Don't-
like,
Don't
no.
fool us.
Don't fool us.
I'll put it in the notes. Look at it. My favorite is the data input of weird tube sponge, another odd sponge.
Yeah.
Mine, I think I put was Sierra said maybe a sponge.
Neat.
I'm not the biologist, so.
That's when you say demo sponge and call it a day. Yeah, sponges are another one that you need genetics.
Look at those
or
colors in the
spicules.
crevices right there.
Yeah, it's neat that it's more preferential green on the bottom of those crevices, or the wall, that overhang.
So
We're
these
definitely
urchins
getting
seem
light.
to live at various depths.
They do. We've been seeing them for a while now.
Yeah.
Oh,
Wow, look at that
We got
cliff.
some fishy friends in Atalanta Cam.
Oh, there's a fishy
Oh, good.
down on our cam too. Oh, there we go.
That's a pretty one.
this is
I'm sure Sean knows what it is.
Can I go?
Alrighty.
Are these the same fish as yesterday?
On Atalanta.
I'm not going to say that. I said that yesterday, and then I was made fun of.
When you asked if it was the same
Saying
fish?
Oh, they thought they were the same fish from 2 days ago following us
Ooh, we
along
got
the
a
Mariana.
loopy in
Well,
the
I
tether.
don't mean.
They're the
Oh,
the
wow,
jack-
look at how many urchins are right there to the
Oh,
right.
What
going
a
on?
little.
It's
What's going on there?
What is a
All right,
pack of
let me
urchins
drive that out.
called?
Urchins.
Ooh.
A prickle.
Come on.
Did you see off to the right that
I
there
saw
were
those.
a huge amount of urchins?
Yeah, we're gonna work on a
I
tether
got a little tether
situation
man or
real
something
quick.
to do.
Okay,
So they're
sorry. Thank you.
Gonna, let's see, what is a
Just,
group of
you can keep
urchins
going up the
called?
hill there, Annabelle. Just watch your
Roger.
distance off the hill.
Oh,
Coming up the hill.
Yeah, I got
did you get one?
a loop.
Yeah.
This is a herd.
Current just folded it over on itself, I think.
Yeah, I think
What
you're
is a
right.
group of urchins called? What is our collective noun?
This is a herd.
A herd of urchins?
Yeah.
Sure,
I
okay.
think of a herd as like something that could come running through, and I don't think urchins quite fit the bill.
Yeah,
Nice.
What
I
do you
think it's
mean?
being ironic.
Oh, nice.
You don't
There it
think they're
goes.
all going to get up and
Good
start
job, guys.
Good catch.
going
Yeah,
across?
they're coming at us. Get out of the way.
I'm looking at these.
All right, gonna keep coming up.
Oh, I
Yep.
found the list
you.
of all them.
We are schmoovin'.
Look at that Atalanta view.
Woo!
Yeah, and if I'm correct, there's no lights on, right? Or
I have the lowers on right now, but I could turn them off. Da-da-da-da.
Oh,
Did you open the iris a little?
Annabelle, keep winching up.
Yep.
Watch the terrain.
so pretty much everything on Atalanta right now is daylight.
Wow. Sorry to be pulling you.
Doesn't matter, just keep off the terrain there.
Roger.
There you
Nice.
go.
Looks like we're pretty clear. Wow, the Atacam has become the Hercam.
Pretty bright, eh?
Yeah, with like visual on the rock.
Whoa.
We've got the lights turned off, so I think that's just sunlight.
Wow.
Wow.
Stellar.
130 meters water depth.
The crux.
Okay, stop coming up.
Roger.
They don't like our traffic website. It's making sure we're not some automated search.
Okay, I have some great knowledge, and I have...
To move your mic a little closer, Shannon.
Yeah, we can.
We're gonna bear
Sorry.
035.
Okay, I have some
035,
great
Roger.
knowledge from
035.
someone on the chat,
I'm getting
and
better
I
at reading
did just Google
everyone's
it.
faces.
What is a group
What
of
do you
sea
need?
cucumbers called?
I don't know.
A pickle?
A pickle.
A pickle.
All right.
It really is a pickle.
That's awesome.
Cute.
Wow.
I think
Look
they
at the
should be
chart deck.
called sea pickles in
Oh,
general
wow.
because they are in salty conditions.
Tilt your camera all the way down.
They have been
All the way
pickled
down.
Yes.
in a brine.
Yes. AKA the salt water.
Yes. So then there'd be a
Looking
group of
good.
them would be a
Okay.
cucumber.
Wow, it's really brightened up.
Look at that.
There's Herc in the Atalanta view.
Herc looks
Close the iris
gorgeous.
just
Glowing.
a little bit.
Yeah, sure. Oh, that looks lovely.
These are more rocky reef areas. See a lot more color.
Is that moving?
Yeah, there's a fish.
Oh, it's a
down
fish.
there, yeah.
Okay.
He just
It looks
swam
like an
behind the
eel
rock.
for a second. Annabelle, someone wanted to know if it got caught in a train or did it just get a loop with a tether?
We were kind of on a cliff there, so we're just, see there's a bit of a plateau happening right now, so we had to up and over, and while we were looking at that, there was a loop, so we undid the loop, came over the top, and now we're here.
Thank you for
Great work.
answering that.
Sometimes if there's a current like coming hard from the side, it's not really a turn and it'll just fold it over on itself. But you can still pull it that way, so happens.
Big props to Mike for
Yeah, y'all
making that happen.
handled that.
Oh, you caught
Annabelle,
it.
you caught it.
Yep.
Oh, thank you.
Fishy.
Oh, what's that purple up there?
Some kind of
Algae
algae or
or
reef forming. We're kind of getting more in the photic zone here, so
Now it's like a fish
expect
tank.
to see more reefs.
Yeah.
So
look
for
at
the
'em all.
silliness, this woman, or ma'am,
Oh.
sorry, that would like to do a Seabiscuit sticker with a jockey, they would like to know what we would want that jockey. She was thinking a squat lobster, but wanted to know if anyone had a more appropriate idea.
I
Can-
feel like
Sounds pretty good.
Oh, there's something
That
on the left. there. I
Atalanta
don't see what it
fishy.
was. Yeah.
Hey, yeah, I think your microphone has gotten away from you again.
Oh no, hello?
There you go.
Atalanta fishy.
Oh yeah.
Keeps drifting.
Atalanta camera is just
beautiful right
How
now. It's
how
so
stable is the environment right now? We
There we go.
Oh, look at
dealing with
that.
currents still, or
pretty.
No, it's
Whoa.
not bad right here.
Little
Okay.
Up a
Okay.
little, Annabelle.
triggerfish
Look at
Roger,
the tail
coming
on
on it.
that.
hanging out by our
could we?
tender.
Oh yeah.
This is
It's
gorgeous.
kind of interesting. Just this. Is that a coral
It could
or
be
is
old
it
framework.
old framework?
Would you like me to keep it
Zoom
around
in video?
15?
Let's get a look at some of these
Generally, but if
gorgeous
we're close and you
colors.
see the tether flying up like that in front of the
Roger.
camera, just come up a bit.
Thank you.
Oh, this is so pretty.
You know, this color and the rubble and
Some little
everything
fish hiding
is very close to a lot of our areas in Guam that I dive.
yeah.
This is, you know, one of the areas that we'd go over and would be kind of dead and we'd move on from, but it is indicative of what some of the higher corals look like.
Yeah, there's some, I think the little yellow branching up might be types of sponge.
Sponges.
What's the blue? Is that a fish?
Yeah, there's blue up in another spot
I
too.
don't know if it's a fish or just the color. It looks like a flower garden almost with all the cool colors.
Yeah,
Zoom
it
in there.
does.
There's like so much for me to see.
It's so gorgeous.
What
I
is
think that just might be the shadows from the sun, actually.
the blue?
Yeah, I think it's like, because it's the sunlight's coming through here, right? So
I think you're right.
'cause
It's amazing how deep the sunlight can go.
I'm
It is
going
a
to
weird
highlight
blue color.
Atalanta
It is.
right
Oh, I-
now.
Well, if you go outside, it
Coming
is
out.
a very sunny day.
Yeah. Yeah, succulent
Annabelle,
greens.
turn the, see the gain slider on that sonar?
Whoa, down here?
Yeah, turn that down.
There's a little
This-
fish chilling right there. There it goes.
Nice. Oh, I cannot see.
It's okay. Here, let me tilt it for
Oh,
you.
thank you.
He is watching us.
There. Oh,
Whenever you were kind of touching that white arrow, it moved down for you.
Oh, hello.
Oh, gorgeous.
Gone down 10% now.
They are chasing the lasers.
Way down.
Yeah, look at them.
So,
more?
all the way.
All the way.
They're like little
That's 1%.
cats.
All right.
Nice.
Thank you.
Still super loud.
The range is pretty low, too.
Wow. That is interesting to look at, Isabella.
Yeah, the Tritech.
What is all of
Do
the
you want
orange,
me to
the
fiddle
cliff?
with the range?
It's just hard substrate. It's so
Hard
close to it.
substrate, yeah. Picking up all these.
Sorry, Annabelle, what'd you say?
Oh, I was just offering if you'd like me to fiddle with the range, if that would help.
No, leave the range alone. That's okay.
All right.
Got lots of little hidey holes again with the cracks.
A little school of fish. Lots of little ones.
More
Oh,
of those
yeah.
jacks. Oh, that's what they
A little bit of winch weirdness. Seems slightly improved, maybe?
Yeah, it's a lot better
Gorgeous
than it
colors.
was.
still looks
Not
like
great,
a.
though.
Yeah.
Were they
I
angelfish?
tried to be careful when I was
Alright.
wrapping it back on.
Yeah.
Oh, the ones back here, the black and white striped
Mhm.
ones. They're the ones from Finding Nemo.
Willem Dafoe fish.
William Dafoe.
And is that a? I never want to say it. It looks like a shark, all the way up there, but it's kind of hard to see.
I'm not spotting it right now.
I'll
but
keep an eye out on
it.
Adelcam.
It's up on the top. You can see its tail kind of
Ooh.
on the right side
Burger rat.
over there.
Yeah.
Maybe we can get it in the frame in a bit.
So someone asked if I've ever taken a cat toy laser down with scuba diving. Actually, they make ones for underwater so that dive guides or people diving together can point out things easier than with like a large flashlight.
Fish in the
So
hole.
yeah, they do actually make those.
Whoa, look
They
at
also
all
make
of
UV
these.
flashlights.
They're
Ooh,
gorgeous. And
this
the
is
little-
incredible.
oh. Yeah, we're starting to get some more relief.
Wow.
I missed that highlight.
Because you were oohing and awing.
Yeah. That's okay. I see so many fish and schools of fish in here. We're good. Thank
Can
you.
we look over to the right for a little bit? I wasn't sure if it was a...
Yep.
See
That
if
looks
I
like
can
a
get
Porites
along.
colony.
Yeah, it's up on the top right corner. I don't know
That
if you
looks
guys are
like
seeing it
coral to me.
move as well.
Top
In,
right
Zeus?
corner.
I'm not seeing it.
Or
I'm not
Atoll?
either.
It's on the Herc camera.
Oh,
What are you
okay.
looking at?
If you pan more right, maybe, there was a larger fish moving.
Oh,
Yeah,
he
these
is right
things.
over there. He's kind of hiding back there.
Yeah.
Oh, there's a couple of them.
Oh,
Oh, look at that.
Oh, now we
yeah,
get
Little
more.
shark.
White tip.
Fish highway.
Nice.
Someone just said there was a white tip?
Yeah, I just
Yeah.
saw a
the
white
ones
tip.
going
Saw it.
by.
Smaller.
It's like right in the background over there.
Yeah.
Now I'll be curious as we get up
Where's
closer to the
it?
top if it's going to be a reef
This
white tip
guy.
or an oceanic. Because we're at 200 meters. Oh, 112 meters. So yeah.
We're really shallow. Gorgeous. Look at that guy. So many fish.
Little bit of terrain on my
I need
20.
to pull up the
Really mild.
poster the other day of all the reef fish. There's some more.
This is where we're going to need Sean to start IDing some of them.
Yeah, I'm trying to get my...
The door just opened. Was
it
that
might be
him?
Sean.
Nope.
No, I'm trying to get my food fish up.
Yeah.
My computer has decided to not work.
Yeah, we've got these big kind of coral groups right here starting to show
Looks
up
good
too.
now.
Yeah, everything's looking swell.
Roger. Swell.
What are those dark things?
Dandy.
On the top.
Might be coral.
Yeah,
Is that another
I think coral.
big sea star on the right?
Where?
The flatter.
Is this one?
The spotty
Oh,
one?
good spot.
Oh, nice. Yeah. Hey, zoom in there.
Got it.
Oh,
Nice. Yeah. Actually getting reef coral. Oh, look at
Nice.
this.
Yeah, that's a coral. These are porites.
Wow.
It did
I
kind of
like
look
the
like one
little
of those flat
fish.
little pancake stars, though.
What is it called?
It's a coral.
But you
Are
said
those
porites?
tube worms or what are the circle
Oh,
ones
it's
with?
circle of porites, so it's like the pores. It's not a species. Sorry, I don't know what species this one is.
What is the red and yellow? Are those tube worms or are those a type of coral barnacle?
These guys?
Yeah.
This might be a type of sponge, potentially.
Oh, there's a lot more of them than I thought.
That might be an acanthastria coral.
The yellow or the big one? We're
The big
looking at?
one.
Gotcha.
Yeah.
So much purple and yellow.
Yeah.
Okay, full wide.
All right.
There's another little fish around the corner over there. They're watching us. They're like, what's going on?
And is the purple and yellow
Oh, there's a starfish.
related to the wavelengths of light that get down at this depth?
Right there.
Or is it just purple and yellow?
It's a good question. I'm not sure. I don't know if there's a advantage of being this color. Some of the fish seem to be the color, which makes me think that the fish have evolved to look like the coral.
For, yeah, camouflage. Or,
Protective purposes.
Mhm.
Yeah. Some fish friends.
Yeah, I think that might have been some sort of Acanthostria species.
Zoom in there.
Got it.
That might be one of our varieties.
No time to stop, unfortunately.
Yeah. Up in the shallower areas, we'll get, oh, that has the kind of golf ball texture, though.
Okay, full wide.
Coming up.
You okay?
Okay.
No.
Slope's kind of peeking in.
Yep. Coming up. Thank you, Nav.
Always.
There, you can really see the light shining in now.
We're at 95 meters.
Woo!
Closing out on the tires.
Just see it drop off there on the right side.
Amazing how we found some fish and some coral there for a little bit, and now there's not much, and we've only been, what, 20, 30 meters?
Yeah, it doesn't take long to move into a different kind of abundance of coral.
Something's moving up there.
Some more fish.
It's more sediment.
Look at this little guy right there.
Another jack. And then off to the right.
Yeah.
Zoom in there.
Look at him.
Yeah.
Is that a puffer?
Oh.
Yeah.
Yes, it
Oh, he's
is.
pretty.
Oh, he's so, I'm going to say it, he's so cutie.
He is so cute.
And I like he's got all his fins are going.
Yeah.
I love
Wow.
puffer fish.
Oh, he's working so hard. Oh.
Dashing polka dots.
Yeah.
It's
Dashing.
very stylish.
An
It is a very
octopus
dashing fish.
and a puffer?
We're doing it.
There's something, oh, that might just be another piece of coral on the left-hand side.
Or a really fat sea cucumber.
Really
That's what I was thinking.
I think that was a black spotted puffer, maybe.
Black spotted puffer.
Oh yeah.
More of the jack. Snapper.
Thanks, Kelsey.
Yeah.
Yeah.
Quick with it.
She got this reef
Boom,
stuff.
boom, boom.
It's almost like she's an expert.
84 meters water depth.
Something like really far right there. That might be old.
Yeah, what is that? That looks a little...
Might be
I think it's an
coral.
overhang, yeah.
Oh, okay. You know, I kind of feel like in these little overhangs that we have, when I go scuba diving, this is where we
Zoom in.
find the turtles.
Got it.
And I know we're way too deep for it, but...
You just said the T word.
Oh, am I not supposed to say the T word?
No, but I'd really like to see one.
Oh, it would be amazing.
More of those fusellar.
How far down
Okay,
are
full
we?
wide.
We're
84
coming up.
meters.
We're okay.
I guess
Pilots,
That
we're
isn't-
heading
Leatherback
060.
dive
it
this
is
year.
possible.
Zero?
Roger.
Yes.
Roger.
Sometimes the turtles will also go to the cleaning
Annabelle, you got
station.
16
Yeah.
more
Yes,
meters
they
to
do.
come up on Atalanta, and then that's it.
We're also starting
Roger.
to see some
50's good.
Astrophora and
50
turbinaria.
and no more.
That's
What's that?
cool. And more types of coral.
Good
Okay.
catch.
Once you start coming up, will we be doing tail to tail?
If we go up to 50.
Roger.
Sure, yeah.
Okay.
Someone said all pufferfish are cute. Sierra, it's a prerequisite.
Amen. I
Closing
agree.
the iris a little bit.
I really like, we get some in the bay in Pensacola is burrfish. They're a type of
Oh, there's
porcupine
something
fish.
black sticking out on the bottom
Oh
left.
man, that tether's a mess again.
Oh,
Yeah, I think
Cucumber?
that might be a
That's the
cucumber.
that we were saying was
Yeah.
pretty
Run
big.
that out the
Oh, I
other
thought
way.
I had seen another like cleaning station thing.
That looked more like our Belati
okay.
that we have, just a lot longer.
loopies. It's popping out.
I love this drop off. That is such a cool perspective. And then to stare out into the void.
Oh, jeez.
Nice. Good job.
Thank you.
And then the void stares back.
And then the void stares back.
Can we try to find that?
Keep that stretched out a little bit
So
more,
we'll
Annabelle. The
see
current is just hitting it
cucumber.
from the side.
Roger. I'll pull up.
Yeah.
We stop at 50, right?
Yes.
Roger.
As we pull away, it's a good example to show the importance of white balancing, which we do earlier. But if we didn't white balance, that view of everything just being kind of blue and brown is what we would be seeing in the photic zone, simply because of how light passes through the water column.
I'll keep an eye on that tether.
Ooh.
We would see it as a lot of red.
Wow.
Look at these
there.
coral.
Yeah. Oh
Zoom in
man,
there.
this looks like a lobata.
Lobata coral. Is that brain type of brain or different?
Wow. Going all the way.
Oh no. Oh, interesting. It's actually a soft coral. And then there's a heliopora to the left, the hard coral, but the coral on the right is a soft coral, which is out of my,
No, you're good.
I don't know that one. But it looks like heliopora off to the left.
And we have some reef fish in there.
Yeah.
Trying to eat the
Awesome. That's
lasers.
gorgeous.
Probably be far more valuable if I had learned all of my reef
Going all the
fish.
way.
It takes so much memorization, and then if you don't use it, you do lose it. Like I am rusty on my fish because I haven't been doing benthic surveys for like a year and a half now. Okay, this
So
is
at one of the beaches in Guam that we dive at quite frequently, we see something very similar to this, and we just unscientifically call it our bubble coral.
It's like bubble tea. Bubble
Yes.
technical bubble coral is like it has bigger bubbles like what we're seeing up there to the right. That
Yes.
is-
Oh,
yeah.
Oh, yes, that is far
So-
more like the bubble coral.
Yeah, so I don't know if we actually have two separate species, but what is colloquially known as bubble coral are those in kind of the center of the screen right now.
Oh, this is just gorgeous.
Yeah, it's absolutely beautiful.
Wow.
And if we can zoom in on the bigger lobes of bubble coral, sometimes there's really cool shrimp in there.
Oh, yeah.
Go ahead and zoom in there. These ones?
Got it?
Yes.
Yes.
Oh, it looks like something on the.
Looks like a
Right.
big, healthy one.
Wow.
So these coral aren't heart corals, so they're not scleractinians.
Yes.
Could it be possible that these are actually the same type that we have in Guam?
Yeah.
Because anyone that's listening, if you go to Outhouse Beach, to the left at 60 feet, that's where our bubble coral sits.
Oh, hello.
Oh. It's gorgeous.
Very.
Oh, the texture is just so nice.
Oh, I'm
It's very soothing.
Yeah.
It does look like bubbles.
I don't see any shrimp or anything
Me
fun in
neither.
it.
It's creating some nice habitat for fish.
They probably hide in this like anemones. I just want to poke it.
I want to like rub my hands through it.
Yeah.
Wonder if it would sting.
Yeah.
That is surface lighting, am I correct, Annabelle? Atalanta doesn't have lights on? You have to move your microphone closer.
I was muted.
Oh, okay.
Hello. Yeah, this is all surface lighting here.
Awesome.
I had the lower lights on, but honestly, they weren't making much of a difference.
Look at all these different kinds of reef fish.
Wow.
Wow.
Squirrel, this is amazing.
This is gorgeous. And there's some like other kinds of coral like over here
57?
mixed in.
On deck.
Roger.
Wow.
Zoom in there.
This is just
Got it.
stunning.
This is gorgeous. So we're about 78 meters right now?
Almost 80, 70 to 80. Going back and forth.
You've got all these mixes of coral. You've got more of these sheep kind. You've got the softer kind. Just all mixed in here. Gorgeous.
Yeah, so
Wow.
this formation that we're seeing, that kind of round plating, that's a foliose structure. It's a squirrel fish, I
Squirrelfish.
believe.
Oh,
Yeah.
he's looking at us.
Oh.
Yeah.
It's just
Squirrel.
like a big field of it, too.
It's like art. It literally looks like a piece of
Yeah.
art
Oh.
hanging on the wall.
Ooh.
If I were a fish, I would live in there too.
Me too. What a great house for a fish. Like, talk about real estate. This is
Location, location, location.
What is?
We
Whoa.
might, yeah, we've got our unnamed seamount. Apparently
Wow.
it's oasis.
perfect. Oasis of diversity and
Wow.
structures and
Just
colors.
doing a little pan, and look at how much
Wow.
on Atalanta. It's just fields and fields of this coral.
It all just looks like one giant, like, blob all together.
Wow.
This is beautiful. Yeah, and just like, what, an hour or two ago? Completely different.
Marin, is there anything wrong with the video? Or should I send in another?
No.
Michael reported a minute ago that it should be resolved now. It was a Starlink outage.
Well, I'm getting
Thank you.
getting some messages on the science chat that's still choppy, 90% pixelized.
Oh
But that
no.
just came in a couple minutes ago.
Yeah, this
We'll
just
see if
came
it
in the last 2 minutes for me.
Yeah, so-
Everyone said it was doing well.
Yeah, it did come back, and then maybe it's gone again.
Yeah.
So I think this bubble coral that we're looking at, Plerogyra, P-L-E-R-O-G-Y-A, is the genus.
Do a partial zoom.
The polyps are described as grape-like vesicles that are in clusters. It may be tubular, which we're seeing if you look down at the framework, that are in clusters, irregularly shaped depending on the state of inflation.
Ooh.
Vesicles are extended during the day and may retract slowly if disturbed. This is an insane, I have never seen anything
Yeah,
like this.
Now that you say it, it does look a lot like grapes.
Yeah.
If you
It's
like
look
a
on
whole bunch.
Atalanta Cam, you can see that one like spot of the
Yeah.
sheeted coral. Is that what you were calling it?
Yeah.
It's a big
To
line.
the right.
It's like, that's cool on the Atalanta cam on the
Yeah, there's
on
a
the left. It's like a-
trough cut there.
Like a separation almost.
yeah, I wonder if it's like a fracture that's got some more sediment in it, or it does just changing the substrate.
I can only imagine like the network of fish that just swim underneath all these.
How much we are not seeing.
Yeah.
Like you can see all the little ones
The little
in there.
purple.
And
Should I come up?
It's like a
We're at
field of flowers.
55-ish.
You come to 50.
All right,
You can see some
I'm gonna pull up the last
breakage.
5 meters here.
Roger.
These
Okay, I'm
tubes
coming
right
out.
here, is this the underneath part?
Yeah.
Copy.
is that a bunch of it dead in there?
Yeah, but honestly, this is a really healthy pop. I mean, this is
This looks really
this is
good.
insane. This is
keeps
really
going.
special
Wow.
to
With
see
the Atlantic
corals.
camera, you just see
All
the
right,
expanse of
that's
it
50.
too. It just goes
Yeah.
on and
Roger.
on.
It's
Yeah.
really
Let me try and
pretty
get a pan of
wild
it.
to see this level of coverage.
Yeah, Isabella, don't move us
And
when
what
you do
was
your
the
next
name
move.
of
Don't move us
these
upslope
coral?
anymore. Take us
I'm thinking it
across
is a
it or slightly
Pleuriogyra
down.
species.
Down. Take.
Pleuriogyra is the genus.
Yeah.
Okay.
So yeah, just when we make the move, make them just ever slightly down slope.
It'll all have to be 90 degrees and lower.
Wow, Atalanta cam, I just highlighted that too. Just look at how
Going
it
over
seems like
these,
we could be
yeah.
over
Close
this
it a
for
little bit.
a long time. It's amazing.
Like just the pan there.
Someone says it looks like a field of purple broccoli to me.
Yeah,
Broccoli.
I was thinking cauliflower.
Yeah.
Cauliflower.
There's
Those are
something big
pretty
in the
big
back
fish.
there,
Yeah.
It's kind of wagging his tail.
Yeah, the tail movement is what I look for. The more exaggerated.
It's just so gorgeous. It's
There's
like a
some
field
piece
of
of
flowers.
coral or rock down to the right.
Okay, transect time.
Yeah.
Yeah.
No.
Honestly.
You would have to almost do just like percent
Yeah.
cover
Yeah.
at this
Yeah.
point.
Yeah.
I think someone being handed a transect of this and having to count all of these would be kind of...
So what you do when you have this much coral cover and you're trying to do a transect is we would define starting point A to B. We would have a transect reel. This is if you were a diver. And you would lay out the line. Let's say we're doing a 50-meter transect. And then you would do a 1-meter square every 5 meters of that 50-meter transect on alternating sides would be one way to handle this much coverage.
Tether's a bit loose behind us.
Yeah.
We
pull
have
it out.
that foliose coral down below, and I think that is a
Roger, do you want me to tail the tail with you?
Leptoria
Second.
genus.
Okay.
Yeah, I've also seen people where if there's so much coral coverage, they have to like do almost like a point system where they like put random dots on a screen and then just like characterize each of the points.
Yeah, actually it could be a Montipora. It's hard to see when you can't, of those folios, there's a lot of different corals that actually make that round circular sheeting. And so if you aren't able to see the actual polyps, then it's quite difficult to get it down to a species level and sometimes
Okay,
even
yeah,
a genus
flip it around
level.
Roger.
But perhaps a Montipora.
I'm going to flip in the positive direction. And tail to tail.
Someone asked if you think that this is a healthy reef.
Yeah,
It looks
I
really
would say
healthy.
so.
I definitely say so.
It's kind of out here. It's an unnamed, kind of area. So maybe not a lot of people have been over here. Not a lot of activity.
Oh, well.
So we got a nice
I'll
little
put a turn in
thriving
there.
system
Hold on.
here.
Yeah,
These corals
untouched.
different color?
All right.
Or is it just the lighting
stay put.
changing of
It's
it?
just the lighting change. So if we're gonna stay in this shallow area for longer, would it be beneficial to do another white balance?
I was just thinking that.
Yeah.
Yeah, someone...
We can take a moment.
Okay.
And then science, it doesn't seem like we're going to go any further upslope just due to the winch payout for ADA.
Yep.
So kind of going,
Follow the contour.
yeah.
Just see if we enter different reef
Sounds lovely.
systems.
Yeah, we've
or
got marine snow again.
I don't know what this is. It's a lot.
Or is it kick up from
Yeah,
Herc?
oops.
So we're starting to see some different diversity.
Oh, shark.
Wow.
Oh,
Neat. Someone get that.
Oh, now that is
it's
a nurse shark.
nurse shark. Oh,
Yeah.
Oh,
my
nice.
goodness. What
Look at that silver color on
It
it.
is a male nurse shark.
You saw the claspers?
Sure did. Got up close and personal.
Did we want a white balance?
Just stretching his tether out here.
Tether.
Okay.
And I'll do
Another,
it over here.
that's a white tip reef shark off in the distance.
Further down.
Nice.
Where?
Oh.
In
I just went by.
the top third of the camera lens off to the right, it's swimming down. It's going to
Oh,
go
yeah, I
over
see
that
it.
foliose patch. Yeah.
Am I just not?
That's awesome. So many different kinds of just fish and coral and sharks and
I'm ready to do the
everything.
white balance whenever you have time, Michael.
Yeah, I'll do it here.
Awesome.
Come up a little bit.
We've got some really cool branching corals. And then off in the distance, you can actually see some different fish assemblage. So as everybody knows, fish swim quite well. So when you're doing fish counts, oftentimes the person who is doing the fish count will go ahead of the rest of the group. And they'll stay kind of stationary and they'll do a survey and count all the fish and then move along. So, oh wow.
It's back.
Oh,
Look at that. Incredible.
Yeah, that is a male.
He's very curious. He's just kind of doing circles around us.
he
What's
could
that?
stick around with us. He's
Yeah.
kind of cool to have there.
I wonder what the Atalanta cam view is on that.
What'd you say, Annabelle?
Should I turn off auto head so you could properly pull me?
Yes, turn it off, yeah.
Roger.
That's
Oh,
So
Yeah.
exciting.
Yeah,
when we drop things.
Come back around.
Also, if you guys are moving your headset around, just mute your mic because it's very loud in my ears.
The poor video team with their really sensitive headsets over there.
So it looks like we're past that field of bubble coral or Pleuriogyra. Some isopora off to our right.
So here's a question that you might be able to answer. So these coral, do they kind of also outcompete one another for space?
Well, yes, they do. So space on the reef is definitely a consideration. And so as you can see with these, what I am assuming to be Pleuriogyra colonies, they have outcompeted all of the other hard scalactarian corals. So in a sense, they have kind of dominated the reef there, but if we go past this patch, then we're able to see kind of a little bit more diversity.
Right.
Yeah.
Yeah, I'm just kind of thinking of like, you know, like trees and different, if they're overarching and they're covering, there can't really
Thank
be
you.
anything growing underneath.
Yes, that is absolutely a consideration.
Over stories, I guess.
Food availability, light availability, space,
It's gonna go black again for a second.
current.
Okay. All right, I'm done.
Okay. Full wide.
I think there's a little bit of a difference.
Yeah, it looks a little bit better. Just so much light coming in now.
You want to keep moving to the east? So
Yes.
where you left the bubble coral to the east, there's kind of a pretty stark line. And then it looked like we went into more fish. And
Yeah.
Are Hercules lights all the way on still?
Yep.
Yeah.
Yeah, that's what it'd be off.
That's off.
Oh, wow.
That's awesome.
That is amazing.
how clear it is that all the sunlight can come through. And
You just made everyone on the feed so happy. They always ask for Hertz lights to go off.
Now they know what it looks like. But this, keep in mind, is at 70 about meters.
Yeah, it is very shallow.
If we did this where we were at earlier, you wouldn't be seeing a whole lot.
No. Thank you for that.
Thank you, yeah.
You're really pretty.
What is that red thing right there? Is it just a
It's a coral.
coral?
Or maybe a sponge.
Yeah,
Can't
there's
really
some
see
like
it.
boring sponges.
Go ahead and zoom in.
But I can't.
Got it.
The orange is a sponge.
Yeah.
And that, to the left of it, actually looks like a colony of Heliopora. And then right below it looks like an Isopora colony. And it's like kind of columnar formation.
Yeah, these sponges have the larger kind of holes, it looks like, that are not really structured. But
Now that one might be, I'm not very familiar with my sponges, but that one might, I wouldn't touch it. That might be a fire sponge, so it has all the nematicists and little stingy cells.
Okay, full one.
Those
The
are quite
orange one is a
painful.
fire sponge?
The orange.
Ah,
no touchy.
Yeah, no, exactly.
We're bearing 0,
nature has a funny way of warning us.
Yeah. Yeah, that bright color. Don't touch me.
Oh,
Yeah.
can I see the fish go into that?
Yeah.
Oh.
Let's be, oh, that's,
When things have a bright color that is supposed to be warning you of danger, it's called aposematism. It's hard to say, but APOSETISM.
How do you say it again?
I say like aposematism. It's hard for me to say. It's a tough word for me to say.
Aposematism.
Yeah. APOSEMATISM.
better. Oh, better. Not really. It's mostly noise.
Someone's asking how long is this dive today? Right now we have another 4 hours and 45 minutes on the dive. We're supposed to end at 8 o'clock tomorrow standard time.
What will be interesting to see is the transition from day to night.
Yeah.
As we talked about in earlier dives, when there is that diurnal
do you want me to throw
transition,
auto head back on?
the food.
Yeah, you can keep it. You can keep this orientation for now. Just put it on and just stay in this kind of normal mode.
The food
Roger.
moves around in the
Back to normal.
water column, and so then you get a lot more activity. Also, some organisms are way more active at night.
Made a big difference when we were on ahi.
some more of our friends.
Yeah, we've got quite a decent diversity of fish in the background. And a pretty good assemblage of coral in the foreground too. Again, those Heliopora.
They're everywhere.
And the Pleuriogyra, the soft.
All
Oh,
right,
it looks
that's
like we have an
gauges.
anemone maybe off to
Okay.
the left, kind of swaying in the patch.
Oh, yeah.
Yeah. Could we zoom in on that?
Yeah, I was going to say, can we
Yep.
see that?
Thank you very much.
See some more of those butterfly fish.
Okay, go ahead, video.
You may.
Might be able to see a clownfish.
That would be awesome.
That would be awesome.
There's some little fishes like
Oh.
hiding all throughout.
Yeah.
Oh, that's
Doing a full zoom.
gorgeous. Look at it swaying.
Yeah, that's beautiful.
It looks almost like one of those like weeping willow trees where it's just like all folded down and over itself.
It's kind of cool that we sort of sway with it too.
Yeah.
Tether looks all right.
You can definitely see that there's some surge in there.
Which again, that makes sense because that would mean that there's all of this food available for these corals to grow. So
I just love how everything's purple. Like purple is the theme here.
yeah.
They all got the color coordination memo.
085.
Okay, full wide.
Roger.
Right.
All the fish you can just see hovering above in the background there. So
I really like these orange ones.
the orange fish.
Yeah.
I'm wondering if it's a snapper. I'm going to defer to Sean on that ID.
Now we're getting into a different zone.
We're just following this contour here.
One I'm clearly more comfortable in than the deep.
Hey, you know, it's all about being curious.
Yeah, it is.
Expanding your knowledge. It would be so silly just to be an expert in one area. Why not learn about everything?
Yeah.
And we're still in
Decide
about
what
70
makes that tether
meters.
look uglier.
Yep. We got
Those
like
are
half a
saddleback
loopy,
grouper.
and it's good.
With the white
I think
and
it looks
black
better
stripes.
with a positive half turn in it.
Yeah.
Ooh, look at that drop off.
I don't think I consider myself an expert in anything. I'm very much more a jack of all trades.
I know, that's a great way to be, though.
Yeah.
Isabella, what is all of
Yes.
that
Bring your heading
scatter
back
and
around to
bright
starboard.
color down there on your lower screen? This one?
Starboard?
This Tritech? Yeah.
Do you want me to spin positive direction and meet you that way?
Starboard, yeah.
Roger.
To the right.
This is a sonar that we have on the front-facing portion of Herc, and it gives us like an idea of what's in front.
Get that 6-8 wrap out.
But right now we have a lot of substrate, and the gain is really intense, so it kind of looks like a big splatter.
It's pretty. Looks like a night sky.
All right, are you coming up? There you are.
Oh wow, this is awesome down here.
Yeah, I see all the different...
Whoa. That is beautiful. More of that plating sheet structure.
It literally just like is a field. Just all different shapes and sizes of coral.
I'm feeling pretty good about the call to get up to the top quickly.
Yeah.
Yes. Agreed.
I'm going to be very curious if they're going to stay at the top through twilight, or if they're going to make their way down. Or
I suspect they're going to stay.
Yeah.
How many waypoints are there?
We're done with waypoints.
we never went up to 9.
Where it's
It
too
was too
shallow.
shallow.
Oh, okay.
We're at our shallowest, really, within a few meters that we're going to get.
We can't.
Atalanta's at her upper limit right now.
Yeah.
We've got some squirrel fish, some soldier fish, some snapper, a trigger fish.
What?
Come down.
Roger, coming down.
All of this plate coral is what I'm used to diving with.
Yeah. It
Tons of it.
could be a Montipora species. I'd need to see the polyps.
It's just making such good little hidey holes for them all to go under.
I know.
Do you have any shots from here?
It's
That's
doing
pretty...
Yeah,
get some good
that's stunning.
Oh. Underhang. Oh, so much color, so much life.
Oh, there's something kind of underneath the middle plate.
Turn the other way, Annabelle.
Roger, turning around the other way.
Maybe it's just
Just
more
watch
coral.
the turns counter, see?
Just kidding.
Oh, we've got Eva's favorite, the blue striped snapper.
Ooh.
Oh.
Yeah, they're all just hiding under. Coming out to look at the lights.
0.5 in the tether and 0.3 in the 6/8. Both negative.
It's cool how you can see a change of deeper down. We had we still have a lot more invertebrates here too, but a lot more
And
species of
come
fish
down
than
more.
we saw
Roger, coming down.
way earlier in the dive.
A little bit.
Yeah.
Oh.
And so you can see underneath these sheets, there's like a pretty big gap, right? That kind of shelf. And that goes back to what Sierra and I were talking about in terms of light availability. So these sheets are actually growing
Have to
out
leave here,
further,
pretty
trying
far away.
to access more light, and not a lot
There's more here though.
that is photosynthetic is able to grow underneath, and so it does create this really fantastic habitat for all of these fish. Oh my gosh,
Look
this
at
is
those.
insane.
Is that another
Oh
anemone
my
we just
lord.
passed?
Look at in the background.
Wow.
It's
Look
like
at
fish highway.
Fish City, yeah.
Got a good school of fish back there.
It's like the channel they're all taking. They're all going to work or going home.
Yeah. This is how Finding Nemo was developed.
Yeah, honestly.
This
Bring
literally
it down
looks
to
like
15.
Finding Nemo.
Roger.
This is so special to be able to see
I
this.
don't know.
I got some
There's just some
pictures
good
of them,
stuff
yeah.
over here.
Again, we're at the redfish. We've got squirrelfish and soldierfish. The yellowfish with the blue stripes are the blue striped snapper. Wow.
Wow. It
Oh,
just
I
keeps
am in awe.
going.
We are at an unnamed seamount
Unnamed
in
and
the
Yes.
western Mariana Ridge.
An unexplored seamount.
Unexplored.
I have goosebumps.
Yeah,
And
it's
it's not
yeah.
just because the aircon's on.
It's like everywhere you look is life.
This is what a healthy
Oh,
coral
look at all of
ecosystem
that
should look like.
school.
Yeah.
Wow.
Wow.
And what we're seeing at this depth, yes, it is, they are corals that we would also see in shallower waters, but because we're still fairly deep, we're able to get that cooler water. From 2014 to 2017, the Marianas experienced very significant bleaching events due to sea surface temperature anomalies, warmer than usual water. In Saipan alone, we lost over 90% of our branching acroporids.
Wow.
Mm-hmm.
And so the at the shallower zones of the photic area, you're going to get reefs that would have looked like this if we weren't experiencing these types of anomalies. Now, of course, wave action, typhoons, and things like that also play into it, but a lot of it is water temperature dependent.
Right.
Well, we're at 28 degrees Celsius, which is about the normal surface temperature of water in Guam.
Really?
28?
Yeah,
Is that?
82.
Ooh, that sounds nice.
Yeah.
For,
We're usually 27,
I
28.
love these shots here. You can just see them all in the distance.
I think if I keep highlighting, I'm going to be in trouble.
Look at this
Keep
guy
highlighting,
coming
please.
through. Yeah,
Oh,
look
what's
at
that?
guy.
What's the spotty one on the left?
I don't know. He's
Let me
about to
see a
come through.
spotty one.
Right here.
Whoa.
Oh, who are you? Let me find out.
Far
Doing
away.
some circles. Yeah. He's keeping a distance. I'm just blown away.
Yeah.
I have never seen so much life in like one photo, like one screenshot.
Yeah. So, you know, I was saying earlier, we have over
So
376
flip Atalanta around, would you?
Roger. Tail to
types
tail.
of coral in the Marianas. Now that's a bluefin trevally.
Yep.
And so that makes the Marianas, Guam, and the Northern Islands the most have the most coral diversity in the United States.
Wow.
So we have really high coral diversity.
Hold on, just come back. Got this the.
Well, it's cool because you have like almost both extremes. So not only do you have this really high coral diversity, but then you have like the deepest depths in like all of the ocean. It's all in like one not concentrated, but you know, like one given space.
Yeah.
One geographic region.
Yeah.
Little terrain near my 20.
You don't have to go far.
Yeah.
It's kind of the same story with the arc and the back arc and the species at the
May I come
hydrothermal
up on the
vents,
winch?
where you get
Roger.
everything.
I'm just impressed at how much this reminds me of a lot of the scuba diving I do in the Marianas. I mean, so much more abundant, and this would be amazing to see this. Our reefs, sadly, are not this healthy, and so this is just spectacular to see.
All right, depth of 55.
Roger.
Just keeping an eye on that terrain there. I'm practically in the recovery position.
Oh.
Not yet.
Not yet. More exploring to do.
Got more of the saddleback groupers, which I think are really beautiful, going off into the distance. Little fuselars, like kind of tiny pink and yellow ones. Looks like we're leaving the really abundant patch of Pleuriogyra.
It is interesting that it changed so quickly.
Right? Kind of have these giant sprawling areas, and then all of a sudden.
And so it tells you something about this
Okay,
habitat
bring
isn't
Atalanta around
suitable,
again
but
into the.
what?
Just look
Are
at
you,
the slope.
Okay, bring it to the slope, Roger. I'll bring it in the positive direction, starboard-wise. We're gonna go head-to-head.
This slope is pretty intense.
Yeah, it just drops. Can't see the bottom.
Do you want me to come on down? I don't have
no,
much
you
room.
got terrain on your 20.
On your sonar, what range are the lines?
On Atalanta, which is the leftwards one, each ring is 20 meters. On Hercules, which is the rightwards one, each ring is 10 meters.
Okay.
We have some coral rubble.
Ah.
I'm gonna have to hold here while you move.
Interesting. So some sort of disturbance.
Yeah.
We are in a really typhoon-prone area. I don't know if we're shallow enough that this would be impacted by that.
At 80 meters is
Pretty
you're
deep.
probably getting your storm base is. I mean, it depends on the size of the storms, and a typhoon can be pretty bad, but it'd be just at the threshold where I'd be pretty surprised if you got too much storm wave action at this depth.
Yeah.
Do you want me to line up with you?
As would I. So maybe it came from
yeah,
downhill.
face the slope.
Roger.
There's some fish on the cam
Yeah. They're still hanging out.
hanging out.
I'm gonna come back in the negative direction. All All right, I'm running a bearing of about 0.
Yeah, looks good.
Roger.
Yeah, so we're kind of seeing all different sides of the slope.
What's that? Zoom in there.
Got it.
Is that more anemone or coral?
More of the Pleuriogyra.
Oh, it's just really small.
Yeah.
Mm-hmm.
Good spot.
So we can see all that CCA down in the bottom right-hand corner. I think that that is a Turbinaria maybe, or Asciopora. Tough to tell
Okay,
without
full
seeing
wide,
the
please.
polyps. All right.
Get a close look at them.
Getting left behind again.
Oh, and look,
Oh,
more
wow.
fields.
More.
Oh, perfect, because I was going to be like, guys, can we go back?
So it's just that little section then.
Yeah, interesting. More of those saddleback grouper.
Right.
Big old triggerfish was in front of us.
Looks like there was a little rast.
Can we get a reset?
Yeah, we just had a break and then more of these.
Also, you
Thank
can
you.
see the contour,
Yes.
right? Like right below a certain depth, these corals stop growing pretty significantly. So maybe it's just not enough light. Maybe only 10 to 20 meters down.
I feel like we've reached the suburbs.
We've
out of the
left the
city.
city.
Left the city life, now we're in the suburbs. Not to be confused with the hydrothermal suburbs. Those are very different. Different suburbs.
Now we've got some of that orange coming back in. We didn't see as much before. And are those sponges, you think?
Where? The orange?
Yeah, kind of
Yeah.
peeking out.
Yeah.
Those fire sponges, maybe.
Oh,
Ooh,
is that
look
a
at
tang?
yellow, yeah.
Oh,
Just hanging out in the coral.
Do you want me to come up a little?
Keep going.
I have about 5 meters.
There's some more yellowish fish
You're
there
okay
in the
right
back
there
right
for the
corner.
moment.
Okay.
We got
Wanted that one to come out.
Fabulous shots. Ah,
More of that Heliopora sticking up, which is one of my favorite corals. It's blue.
blue.
Doesn't look very blue right now.
That's awesome.
Yeah.
I love how colorful corals are. Like, it's just so gorgeous. It's literally like a piece of art.
So I've got word that the sea biscuit sticker is done.
Oh.
And that they posted
What's
on.
this down
Huh, we have
right
a
there?
meandroid.
Yeah.
Yeah.
Oh yeah.
My brain saw the tangles and at first I was like, oh my gosh, it's an octopus.
I'm still on the hunt for one too.
Yeah, you would.
so
Oh, and
Oh,
then
who's down there in that
Little
left?
eel.
yeah.
Yeah.
Oh, that's
Saw
a
that
different.
too.
We haven't seen much of
Yeah.
that coral here.
It's
No?
a meandroid type formation. I'm trying to see if the polyps are. No, they're not individual. Okay, let me.
You can see little fish eye peeking out. Zoom in a little more.
Ooh, great shot.
Oh, nice. Those are pretty big lobes. So I'm thinking it's not Platygyra. Maybe Lobophilia.
Okay.
Maybe, but Lobophilia
full wide.
generally has
Alright.
individual polyps that
Where'd you
are
guys see
on
the eel?
longer stalks
yeah, the
together.
eel is
Down
left
below us.
side. Right
May-
there. Hooking
maybe
his head out.
Symphilia.
What is it?
Eel.
Oh, can we
And a little
zoom
coral
in?
Zoom in.
home. Yeah.
Alright. Feel better about Symphilia. Oh, hi.
Aw.
Look at his
Sock
eye.
puppet.
He's got a little fishy in there
yeah,
with him
he's sharing
too.
his home with some friends.
Hello. Oh.
And they all want to come out and see us.
Aw.
He's got a little coral home.
Does.
Yeah,
That looks so
sweet digs
cozy in
there.
there.
Oh my goodness. Oh,
Whoa.
thank you.
He has
Okay,
decided
full wide.
he is done with
All
us.
right.
That was cool. Oh, wait.
I'm keeping an eye on the tether. Things look good.
Wow.
That's it.
Oh, he came back out. Cool.
Oh, is he coming all the way out?
He's just coming out partially, yeah. So many eel. That was so awesome.
That was so awesome.
How many times can I say the word cool and awesome in the past hour?
Not enough.
Not enough.
This is our ahi moment
Yeah.
Yes.
for our watch. We
Yeah.
didn't get that.
This is absolutely amazing. So much abundance.
And we did see a little octopus earlier. He didn't stick around for very long. But
We
did
still
see
saw an octopus and an eel and some shark.
I think the nurse shark was really. I love nurse shark.
Yeah,
A pufferfish?
And a
Pufferfish.
puffer. Very true.
And a puffer. Oh, what's in the
Here, come
background
on down.
there?
A big
Coming
old
down.
fish. Man, this slope is crazy. And so I think what is so interesting is just the amount of time
Would you like me to
that
bring
it
it down to about
took
15?
for this community to come together.
Right.
Roger.
Oh, yeah.
Versus, you know,
I've got an eye on that terrain there.
the
then.
inshore reefs and stuff,
Lovely.
like they
I'll,
take so long,
I'll
but
stop
how
if it
quick
comes in too close.
change can happen and completely like affect
There's
the
another
entirety
eel.
of the reef. Yeah.
Or is that
This
our
so
eel?
hard.
So for someone who studies rocks and talks about geologic timescales and how rocks form, can you give me some
There
perspective
it
on
is.
maybe like a coral reef timescale?
Yeah, yeah. I would say this took a couple 100 to 1,000 years, maybe. I don't know. That's really hard for me to say. And I guess on a geologic timescale, that's not that long. But the process of these, you know, if we were thinking about the newer features that we were seeing earlier, the newer volcanic features that we were seeing earlier, with absolutely, you know, nothing on them. And now we're
Now, do you
here.
think I could come a little
and
bit closer?
First, the crustose
Or are we
coralline
getting there?
algae had to come, right, and settle. And then
We're
all of
shifting-
the different
we're shifting
organisms.
starboard,
we're
so I think
shifting
it's about
starboard.
to-
Okay, good to know.
It's
Thank
gonna
you.
it's gonna
It's
start
gonna be
coming
there.
into your
Right.
yeah.
Had to recruit over time. And depending on the depth, I mean,
Oh,
a
wait.
polyp can be an individual single polyp for like years.
Right.
So it would be interesting there are folks that are able to age reefs by taking samples of, especially like bouldering corals.
Like
or-
chlorites.
yeah.
Yeah, there's some people, you can actually look at, like apparently you can determine what the sea temperature was like just based on taking a sample of those hard coral. Like you can look at the different bands.
So
that
kind of
it
like
grows
trees?
over the
Yes.
over the
080?
years, and you can
080.
see
Roger.
We're staying like in the middle of the
different
contour.
temperature. People have used it for
Yeah.
like El Nino, La
Mhm.
Nina to look at temperature fluctuations, and you can determine years and years ago what the sea surface could look like compared to like just taking a small sample, which is really,
Yes.
really cool.
And what is so interesting too is you can take a slice of a coral skeleton and you can actually see when bleaching events have occurred. So the coral tissue will die back, and in the coral skeleton, it'll actually look like a kind of a black mark. And then if the coral is able to regrow its tissue, then it'll grow healthy again, put more skeleton on top of that. And so you're actually able to look at the bleaching events just by slicing a coral and looking
we got
at
some
its
right in the 20
skeleton.
there.
Yeah, that's awesome.
Similar to tree rings.
Tree,
Yeah.
sea events.
So people on the chat are saying
Makes
that the
sense with
name
the
of this
slope.
seamount should be Grape Field.
Back up.
Great
Oh.
field.
Grape Field.
It's very purple. We've got Bill about to pop on and take
We have an
over.
interloper
Well, he's
Alright.
about to get on
Yeah,
his
we're coming
ship.
on for
for a few
Yes.
watch
minutes.
change.
Bill
He was just so
is going to
excited.
take over. It's been lovely taking us from waypoint 1 to waypoint 8, the dark depths to the reef. Have a wonderful remainder of this dive. And I'll see everybody tomorrow, maybe in the back arc, maybe with some hydrothermal vents. Back to fluid sampling.
The 12 to 4 will be here
12 to 4
tomorrow.
will be here. All right, bye everybody.
So we are slowly going to be changing ships over the next 10 to 15 minutes.
I think the Coral Collective has had a great time today.
They've been well fed.
Well fed, lots of coral content today. I don't know how much we'll get of the hydrothermals, so y'all just soak it up right now.
I will definitely be watching this in the lounge to see how this dive progresses with the 4 to 8. Although I will say, I think we went the whole shift till now, and we did not talk about food or cookies.
Woo! Win.
It's a win.
I brought up olives a little bit ago.
Well, we
You
did
what?
talk about grapes.
Yeah, we talked about olives and grapes.
Olive and grapes.
And broccoli.
It was with scientific
broccoli.
reasoning
Yeah. It was scientific talk.
So
to kind of describe the shape of the coral we were seeing.
Exactly. Thank you.
Oh, there might be an eel.
Another one?
Where is it?
Kind of in that center piece right there. It's kind of,
Oh, there he is.
Oh, yeah, yeah.
could be, or fish. I
it
don't
looks
know.
like the same kind.
Zoom in there, video.
Got it.
Poke his little head.
Oh, come back.
I didn't see it.
I didn't see it
Might be
either.
kind of behind that rock face.
Yeah, oh, there it is. On my right.
Oh.
Right here?
Yeah. Oh, hello.
Can I turn those lasers off?
Oh,
Yeah.
what is it?
An eel.
Oh, that is an eel. He's popping his head back out. That's a tiny one.
He says, "Don't disturb me in my home, please. No solicitors."
Okay, well, what?
All right.
Someone asked if we had giraffe talk, and we did shortly, but not very much.
Haven't seen any.
We
Sorry to
haven't seen any.
disappoint.
We saw an octopus, though.
And some shark.
And it was right after the transect, so we got to stop.
The new shark was also such a highlight.
Yeah, I think the nurse shark has
So
been my
pretty.
favorite.
Yeah.
Really, really such a highlight.
Came around more than once. He was so silvery.
We're so lucky to be out here.
I really, really couldn't agree more.
I'm having a great time with y'all.
Oh, man.
We love the 12 to 4.
What? Shannon, can I hand you this?
Yes, is it mine?
Yeah.
Oh, thank you.
Bill just gave it to me for some reason.
Oh, he probably, yeah. So Kelsey's just switching seats to her
Data
regular
logging
shift.
position, yeah.
No, there was a,
Hello.
Hello,
Hello, Kelsey.
Hello.
She's on my right now instead of my left. Who's that guy? Ooh.
We're still sticking around 75 meters.
Yeah,
It's pretty shallow.
right. Shannon, where are people tuning in from this day?
Where are people tuning in from?
Get a what?
Yeah.
United States,
Yeah.
Canada, Australia, United Kingdom, Poland, Japan, India, Sweden, Guam,
Do a little-
Denmark, and Germany.
Oh, oh,
Wow.
bye-bye, Herc.
All
We
over.
have quite a diverse group of people tuning in.
Look at all of these. Ooh.
It's been pretty steady today, people in and out.
Right.
So, and they stuck with us through the time that we went down
Yay.
for a little bit.
There's another eel on the bottom, I think.
Another eel?
Where at?
Oh, we just passed by it.
Oh, gotcha.
Oh, yeah, I think you're right. In that bigger hole with lots of little fish. We have far northern California and Eugene, Oregon.
The west coasts are still
Yep.
awake.
We still have about 5 minutes on shift if you want to do a shout out to where you're from.
Yeah. Bet a lot of East Coasters are now sleeping.
Yeah.
But they could be online, you never know.
Well, we have some Europeans on here, I saw.
Okay, true.
Ooh,
Oh.
got a
Oh, what are those?
trevally.
Those look like trevallies?
Yeah,
Yep.
trevally and then some jack. Same family. Karen, Carangiddae. Sometimes it's easier for me to spell things and say them.
We've got a Vancouver Island. Couple of them,
Soldierfish.
actually.
I believe more of that heliopora. Could we zoom in to the pokey looking coral in the bottom right? Yeah, kind
This
of in the
one here?
columnar.
Where?
Yeah, that one.
Okay. Second.
It's okay if not. We've seen a lot of it.
Go
I'm
ahead
just
and zoom.
curious.
All right.
Yeah. Really tiny polyps. Yep. Okay, cool. Yep, that's
There's
our
like a little
heliopora.
crab or something right there
Oh,
hanging out.
there is.
What's kind of the yellow?
It looks like maybe one of those lionfish one we saw earlier. Off to the left and down just a little bit.
I can never tell because the shape is so like.
Yeah.
Mm-hmm.
It's a great shot. Look at the little tiny little polyps.
So you didn't type in vain. I have, oh, let me go down.
I'm coming out.
We've got some snapper.
California, Canberra, Australia, Whidbey Island, Washington, Sweden, South Australia, Indiana, Midwest US, oh, Missouri, Houston, Texas, Beaverton, Oregon,
Yay, Oregon.
Lewiston, Idaho, Austin, Texas, and Moore, Australia, and Melbourne.
Nice.
And Quebec, Canada.
Okay. Now we're
There's
into
a mining
the full-on
security
watch
agent.
change
Taking
going on.
over.
Yeah.
Yeah.
I'm still chilling. What was the snack for today? What do I have to look forward to?
Oh, it was that pastry
There
thing with
better
the
be some
powdered
left.
sugar. Oh yeah.
I
those-
didn't grab
leave
one.
me
Would it
some?
mind
Go,
bringing?
go.
Oh, I should go.
Yeah.
Okay.
Quick
All
before
right.
the watch change.
I'll have to grab
I'm
me one.
excited now.
Yeah.
Yeah, I can.
There's our food talk.
During the shift change.
We did not talk about the 3 o'clock cookie, but
Yeah.
we've got Go Beavers. That's the second person.
Go Beavs.
That's
Yeah.
yours, right? That's for you.
Oregon State University. Go
And
Beavs.
friend Derek, I'm sorry, Denmark.
Denmark.
There's a city here.
Been
but
a long day.
yeah.
Very true.
The one that I can't pronounce are where the people are from Quebec.
Quebec.
It is in French, and
Yep.
It's very lively in here right now.
Yeah, we're still working on shift changing right now.
So I think it's pretty remarkable how different this seamount is from the one we dove on yesterday. Maybe, do you think it's the side? Because we're up on the south edge of the summit.
What side were we on yesterday?
The northeast.
That's been the kind of thing I've seen
Maybe
in
it was
literature,
like the current direction.
There's been a lot of studies of like whole seamounts where different sides have different communities.
Yeah.
So might be pretty similar here.
Because
how far away are we from yesterday's seamount?
Well, it was about a 7-hour transit south, so that's probably 70 nautical miles, more or less. Yeah, so not super far.
No, but we've just had different communities.
Yeah, totally different, and the abundance is crazy different too.
Yeah.
Like,
Bill, you were in the news this week?
I was. I've been all over the news for
someone says you're doing well considering you're in the middle of the ocean.
Yeah, I know. I've been doing reporter interviews from sea. It's been a little crazy.
Is it because of all the volcanic stuff happening right now?
Yeah, well,
or predicted to happen?
Yeah, the one I study, offshore Oregon, axial seamount, has been in the news a lot.
Is it erupting?
No, but we think it will soon. So, so that's all the interest. Yeah.
Yes.
All right, we're still doing the watch change here.
We have India here.
Pause. So you heard they're still having trouble with the satellite link?
Yes, it has gone in and out. Right now, everyone seems to be doing well. I've had to do, reported a couple of different times.
I think it's been going in and out, it sounds like, so.
Yes, someone said, isn't it the anniversary of Mount St. Helens? Yes, it was just a couple of days ago. We were talking about that on the ship.
May 18th, 1980, 45-year anniversary.
Yeah. You know, we were talking about it on the ship. I'm 45, and somehow I remember it, which can't be true. It must have been sometime in my elementary years that they started doing documentaries or something to make it so poignant in my mind.
Yeah, I'm old enough. I do remember it. Mount St. Helens erupting 45 years ago. Yes, there's a subset of us who were that old. I actually worked there for a couple of years right after the big eruption. Wow. So, yeah, that was a very...
Oh, is this a net? These are nets, huh? Is this
a
little fishing net?
That?
Oh my
is
gosh,
it?
it's a fishing net.
Wow.
Oh my gosh.
Wow. Good eye.
Aaron, do you want to give us a zoom if you get a chance?
Yeah, it's definitely
Are you getting some frame grabs?
Yeah.
A fishing net. So
I saw something
we're not the first ones here.
Wow. And it got caught, so it's pulling super taut.
All right.
At least it's
it
not.
It almost looks like a piece of wood, like it's been pulled, so.
Yeah, and it's obviously been here for quite a while, huh? Wow.
Wow.
These corals look like broccolis.
Yeah, so they're a species, genus of coral from Pleuriogyra. So they've got a tube at the bottom that is calcium carbonate, but then they've got these soft polyps, and we've been calling them grapes. A lot of the viewers think they look like grapes.
Oh, is that the end of it right there?
Yeah.
Yeah, that's what I got hooked on, some Heliopora.
Huh. Oh yeah, the coral is going through the net right there.
So much algae on it.
Wow.
Okay.
Thank goodness it's sort of pulled out into a thin line and it's not covering more of the seafloor. Huh?
Yeah, but this is within the U.S.'s EEZ, so I'm curious as to whose net that is.
Yeah. there's some of these are growing on top of it,
Mm-hmm.
or not through it, huh?
Yeah, you're not going to be able to have a lot of corals growing under it again because of just the light availability. But yeah, as you can see, there's like quite a bit of CCA on it.
Whoa.
Wow.
Look at this view.
Dang.
Wow.
Yeah.
Do you guys need the lasers on or no?
No.
No, not right now.
Okay.
I think this is insane.
This is
Yeah, that's like
broccoli.
100%
Yeah,
coral cover.
purple sprouting
Or
broccoli.
grapes.
Yeah, grapes. I was going to tell you all, but here we
nice
are.
lighting.
It's no lighting.
Is it?
Yeah,
That's
all
natural
lit up.
light?
That's
Yeah.
natural.
Oh, that's awesome.
Could we follow how long this line goes,
Yeah,
this
you wanna
fishing line? And
go
could
to
we
the other end?
Can we mark
unfortunately,
it?
It marks back the other way, so we can. I'll go sit on the top, and maybe Johan can get a.
Yeah,
Oh,
I'll
I
start
see.
bringing this that way, but it also looks like it goes a little shallower.
Oh, okay.
Oh, I see.
Yeah.
So we
Can't follow it.
may not be able to follow it too far.
I'm just curious as to how long this thing is.
We're going to be
Yeah.
really blue unless we have some lights on as a heads up.
You got it.
Oh, butterfly fish. Huh?
Mm-hmm.
Oh, those are raccoon butterfly fish. I know that one from Hawaii.
Well done.
Nice.
They have
You've got the
Do they have
butterfly
spots
fish.
on their...
black stripe
Oh,
along
nice.
over their eyes.
All right, it's right off
See,
my
raccoons are real.
right off my porch there.
I still, still don't believe it.
Oh, okay.
Raccoons are real, confirmed.
Amanda,
Hello.
you're with us.
Half a day.
4 to 8 is now complete. We weren't complete without you.
Oh, I'm so glad. Good morning. I rolled out of bed.
Did you just wake up from a nap?
I did.
See, Amanda was late from a nap. I was late because I was having a shower.
Say again.
I was going to say, Amanda's been doing interactions all evening.
Yeah, half a day. I fell asleep on my alarm, so I got woke up because it was vibrating against my cheek.
Oh, poor Amanda.
But good morning. Good afternoon.
Okay, so nobody had to shake you awake. Your alarm clock eventually did?
Yeah.
I've missed my alarm a few times because the vibration of the ship sort of masks.
Oh, man.
Masks it.
Wow. Amanda,
Yes.
welcome to the top of the seamount.
Oh my goodness.
It's covered with incredible things,
It's
And a
beautiful.
fishnet.
but unfortunately this is a fishing net that got
Oh,
snagged
this-
on
Yeah.
the bottom, in the middle
Oh,
here.
goodness.
But it hasn't done too much damage, I would say, which is good.
Mm-hmm.
So the
Oh, it's really long.
Yeah. I think that one thing that is lost on people is the size of the equipment that is used in commercial fishing.
yeah.
No, definitely.
I recently saw a graphic that was showing one of the larger commercial fishing trawling nets, and it could fit the Eiffel Tower in it.
Oh, goodness.
Yeah.
What?
That's the net
Yeah.
floating there.
Wow.
So any idea what the target of this?
The
This
target
would probably
species
be a
here?
Yeah.
trawler.
Yeah.
Oh, boy, it's really
Look at
messed
that.
up here.
So
Is
what
it-
did you
is
say
it
the
up in
target would have been?
I would say this is probably a trolling net, which
Oh,
would
okay.
mean it would just get everything as it
Yeah.
drags along.
Yeah,
Yeah.
Oh, what a mess at this end. Is it floating?
It,
I think
Yeah.
it's
Yeah.
hooked on something, isn't it?
Well, this might be a float up here.
Yeah, yeah. It
So-
must
Oh
be,
yeah,
huh?
those are the 4 buoys that would
Gonna
have
stay a
marked
little
kind
bit
of the
away from
top
there.
of the trawler net.
Uh-huh.
Aaron, you see that?
Yeah, I see it up there.
So is this what they call like a ghost net? where
Yes,
it's
this
abandoned?
is a ghost net.
Oh.
Yeah, thank you, Amanda. That's a really good point of knowledge is when derelict fishing gear or gear that gets lost or snagged on the bottom, it's just cut loose. And so it's referred to as a ghost net because this is still catching and still impacting the ecosystem. Now this one's pretty securely tamped down to the
Yeah,
bottom, but
yeah.
oftentimes these are still in the water column. And so you
Right.
have your pelagic species, fish, cetaceans,
Aaron, do you want to
and
zoom
they
in a
are
little?
just
So-
caught
so
in them.
these are all floats.
Those are all
Do a
floats,
little
yeah.
scan around.
There's a little puffer fish down there.
The biggest detriment that this has had this will have on this ecosystem is A, covering the space for the corals, but also organisms potentially getting caught in the netting as they're trying to bury themselves into the, trying to find shelter, right? And so then the issue with the netting is, depending on the size of the fish, if it goes into those little square slots, it can't reverse out because it catches its gills or its pectoral